That's all good. And I think I can close that. All right. Full screen that. Okay. So this is a blow up of the microscope, the stage, and the immersion oil lens or the oil objective lens. Oftentimes in other courses, this is a lens you're told not to use, right? But in microbiology, you need to use it, all right? And although you're going to use the other lenses to get into focus, when you're looking at bacteria on a microscope, you really have to get it under oil immersion to be able to see what's really happening. At the lens before, which would be 40x or times it by your oculars 400 power magnification, you can see the bacteria okay, but you definitely see it better when we magnify it 1,000 times. So 100x on here, and again, your oculars 10, making it 1,000 magnification, right? So we can see what's up with the light as we look at the cartoon image over here. The light moving through the slide up into the oil, and it takes all of that light and focuses it, allows it to stay intact right into the lens, right? And when you get that immersion oil lens into position, your first thought is it's gonna slam into the slide. It is very close to the slide. It technically has a little spring load up in there. So if you do get a little too close, it will push it up in there, and you're not usually not going to break the slide. All right. There are some potentials, and usually kind of older microscopes that are maybe not quite calibrated properly, and you could turn it a little too much and you've cracked the slide. But typically the scopes we're dealing with in the labs now, usually it stops before you get to that point in the first place. All right. But it does push up in there, so to prevent it from getting damaged. So the light moves right through and gives you a nice crisp image, better resolution, right? And as we look at this other image without any oil on there, you get some light up in there, all right? But you get a lot of it that doesn't make its way into the lens. And so you're gonna see some stuff, but it's gonna be pretty fuzzy, all right? It's not gonna be a very good image. I can definitely tell when I'm teaching labs and I'm moving around looking in the microscopes of students, I can tell if there's oil on there or not, all right? You can see or not see the bacteria like you really should be, all right? You also, as we see here, you need to be very close, all right? And if you put a drop of oil on there and your lens is clear up above it, not dipping into the oil, you're not in focus in the first place, all right? You're not seeing any sort of bacteria. You might think you're seeing something, and it's probably something on the oculars that you think you're zooming in on, all right? When I'm looking at things through the microscope and I'm really initially trying to find stuff, I'm using my coarse adjustment initially, and then I see a hint of color, and I move to my next lenses and I start fine tuning, and then I get to, the point of 400 to 1,000, and I like to wiggle my slide the littlest bit with my on the stage so that it moves back and forth or up and down one way or the other, so I can see it's moving the littlest bit. And when I'm looking in there, if what I'm seeing isn't moving, I'm not focused in on the slide. Like I said, you might be seeing something on the oculars, dirty lens or something like that, that's making you think you found something, all right? If it's not moving, you need to keep fine tuning, all right? And ensure that you put the oil on there properly, all right? So the dark filled, and we showed those images last time with some of those spirochetes where it looked sort of like they were glowing and the rest of the background was all blacked out, all right? Dark field microscopy uses a regular microscope. We're not going to use dark field microscopy in the lab, but we technically could, all right? So the lens, there is an illumination block that basically blocks out a lot of that light. So it makes it so that the image, the specimen itself, the light goes through it, and you're not going to be staining these guys. It's just put right on there, right? We look at things that have been stained up and use something like this. So you don't need to get bogged down with all of these images that show some of this stuff. We're going to see a handful of them as we move through here, talking different types of microscopes, like electron microscopes, that it's interesting to see some of the details, but as far as me showing you a picture for the exam and having you fill in this stuff, definitely is not necessary. So here's a dark field, a different spirochete, Treponema pallidum. This is the culprit of syphilis. And you can see in this image, little kind of corkscrew looking bacteria. And again, the darked out background. There are other techniques that we might employ, thinking this bacterium right here, where we may use some antibodies against this organism that have a fluorescence tag on there, and they will link up to the bacteria and then they glow in the dark. So it's kind of an immunology and microbiology merged together to make these things be visualized, all right? And I'm pretty sure the very last image in this chapter is that stuff. So phase contrast is another mechanism that we can use, different microscope, where we move through and see some details. And it's better, rather than me going through this stuff, to just move over and show some images. I think that does it a little bit more justice, right? So we're looking at the same stuff, all right? Which I think is also important as we're making some of these comparisons. You should be seeing the same slide, right? That makes a little bit more sense, all right? What we're really looking at is this cell right in the middle, all right? There's another cell down below, and this I would describe as artifact, all right? This is not cellular material. It could be a little a blemish in the slide, like a little nick in the glass, something like that, it looks nice and smooth, but when you zoom in with a microscope, maybe it's not, right? Something like that. It could be other things as well, debris or something like that. If a substance, whatever it is that's been put on our cells, have been stained up, there are also potentials for that stain to cause some issues. Sometimes if it's not applied properly, it will dry out a little bit and crystallize, and you see these beautiful shards of color that are really not what you want to see, right? But they look cool, all right? So in this image, all right, we're looking at a squamous epithelial cell. I was scrolling down to see what are we really looking at. I knew I was looking at a eukaryotic cell because right off the bat, I could see the nucleus, all right, right in there. And it looks pretty big as well, all right? So we're looking at a comparison, regular bright field microscopy, and we haven't stained these things up, all right? But you can see the cell and you can see the nucleus. And then we use the phase contrast, and you can see more details about that cell, kind of ideas, hints about the surface of the cell, if you will. It gives it more of a texture, I feel like, as you look at it, all right? It's kind of, I kind of think of it like, standing outside and looking up at the moon. You can, nice, maybe full moon, and you can see a little bit of craters, and then you bust out a telescope and you can see considerable detail with that, right? Kind of the same idea, where you can see it and you can kind of get some hints of stuff, sort of looks a little bit granular, but over here, you can definitely see it. And some other things pop out a little bit better, like another little organelle right here, maybe a lysosome or something else. Nucleus, somebody else, some other players, maybe little mitochondria, that we can visualize as well. Down below, like I said, it's another one, but we're only getting a fraction of it, but nucleus visible in both, all right? The DIC, oh man. I can't, right now I'm like, I don't remember what that's called. It's always DIC, but it's, it will tell me in 1 second. Where is it? Doesn't matter. I'll get to that. I just scrolled right past it where it showed it on there. Doesn't matter. This microscopy, in this image, we're looking at some fungus, all right? There are all sorts of types of fungi, all right? There are all sorts of types of yeasts, all right? One of those packbacks said something about, you know, why do we use yeast in foodstuffs if they cause infections? It's a completely different type of yeast, right? It's a different player, all right? There are numerous things. There are molds, not this guy, all right? But there are some molds that we potentially utilize. If you eat blue cheese, there's mold in there, all right? There are other things where molds are utilized as well, all right? Some soybeans can get fermented and make a kind of little block of fermented soybeans called tempeh. And tempeh is full of mold. And, you know, there's lots of things where you may see warning labels like, oh, be cautious with this or whatever. The tempeh's got a label on there, like a warning label, but it's more of a disclaimer. And it's like, hey, this product looks moldy because it's supposed to look moldy, right? Don't be put off by it. That mold created this product and is safe for you to eat, right? It makes me feel a little bit uneasy seeing all of that mold on there. I've tried some of it. I thought it was okay, but not something that I was like, oh, that's just delicious. I only bought it one time, right? Any tempeh eaters? You know what I'm talking about? Have you seen it? You can sometimes see it at the supermarket, maybe, I mean, it's in the kind of refrigerator section, maybe by tofu and other stuff is where you might find it, because tofu is another soybean product, but it's not using mold to ferment that stuff. So these guys, we'll touch on some details with fungi in the next chunk of material when we start thinking some of the diversity, but we can see these strands, the little maybe fuzz of that mold. We call these hyphae, and a bunch of it together would be mycelium. So the hyphae might be divided like we see here. We call that septate, or it could just be one big long continuum, aseptate, all right? And then they produce these fruiting bodies, and there's a handful of different names that we give them, but it's basically spores that are generated in there that get released out, settle down back in their environment, and grow up like these filaments, and then reproduce again, all right, with more of these spores. Oftentimes seeing some moldy substance, loaf of bread or mold on an orange or something like that. You can maybe see some black or some greenish pigment in that mold. And a lot of that stuff are the spores. It's the top bits that you're really seeing, all right? And you maybe see some fuzz as well. And those are the hyphae, the filaments of that. So here, and this image is a lot blurry in here, but we've got some fluorescence images of a bacterium, Neisseria gonorrhoeae, the culprit for gonorrhea, all right? It is a gram-negative coccus, and if we gram stained it, they would look pinkish. We're not gram staining here. We're using some fluorescence tags to visualize, all right? So with this, the fluorochrome is the molecule that will glow when exposed to some UV light. And in these instances, we've got an opportunity to do it a couple of different ways, all right? A primary antibody is an antibody that's against that specific thing. So against, in this case, gonorrhea. it may be tagged and it could link up directly, right? Or a secondary antibody is something that needs the first antibody to link, and then it detects that antibody. So it's not detecting the bacterium itself, it's detecting the antibody that linked up against it, all right? When we think about those things, antibodies are produced in animals, us or other things, all right? So it might be that we've got an animal that's been inoculated with gonorrhea and it produces antibodies against it, and we extract those antibodies and we do some biochemistry linking those fluorophores to that antibody, all right? And then you've got a different species, a human potentially, and you could have antibodies produced in the human, and those antibodies would link up and interact, all right? Just depends. And in this case, I kind of messed that up actually. If this was a human antibody linking to it, the animal would be producing antibodies against the human, has nothing to do with the gonorrhea, just the human, and then it detects it, right? So whichever way you go about it, all right? Direct, linked right up. Indirect, you got to have kind of a sandwich where you link them together, all right? So that is what we've got there. This image up above is a type of parasite called, it's a schistosome. Schistosomiasis is the infection. Schistosoma mansi is this specific species. And what we're looking at here is a step in the life cycle of this parasite that would be infective to us. We call this stage of the schistosomes a cercaria, and the cercaria would be produced inside of a snail, and they would leave the snail, they'd be in water, and if we happened to be in the water, they would penetrate into our skin, get into our circulation, and then cause infection, all right? That's really what's kind of happening with them. And they look different once they've made their way into us. This is just how they get in, and then they convert into a different looking player. We'll show some details of that later on, right? Confocal microscopy is a bit different, right? It still has microscopy in the name, but it's not like we think about with regular microscopes where we're using these oculars and zooming in on something. We're still zooming in, but it's very different. The light source, instead of a little light source down below, we use lasers. And the lasers, typically it's in a very large room. They're super cooled lasers that in turn focus this laser energy on the substances, whatever it is we're looking at. So here we've got a biofilm, a potentially mixture of different organisms, all kind of forming a community. In this case, a roof-dwelling cyanobacterium. And we can see handfuls of things within here, right? The cells and the secretions that they're producing that help to kind of get them glued to their surface, right? And, another example of some biofilm might be in your mouth. Plaque on your teeth is a good example of some biofilm formation. So this image showing us some details. I've got one quick story about confocal microscopy that I thought was pretty cool. Like I said, these are very different, right? Something that not everybody, not all sorts of labs have these. Many years back, we took a field trip to a National Institute of Health lab in Montana. Rocky Mountain Labs is what it's called. The US has two big labs, CDC and NIH, that do all sorts of different types of research, right? And there's some locations kind of across the country where we see them. See, the main lab for CDC is in Atlanta, and the main lab for NIH is in Maryland, right? But CDC's got some other satellite places. Colorado, Fort Collins, Colorado's got a CDC facility, and Montana, an NIH facility. Really, I feel like it's for the people on the East Coast to come west and maybe do some fishing or some hiking or something like that. That's what it's real. That's really why they want it, right? Deep down, they're like, that's what we want to do. There's others. That's really not all of it, but part of it I think is accurate. All right. So we're up in this lab and we're seeing all sorts of different things. All right. I showed you that image of the spirochete Borrelia burgdorferi. Willy Bergdorfer was the individual that studied lots of different tick-borne illnesses, but Lyme disease and a few others were definitely what he spent a lot of time focusing on, and in turn named after him, right? He did some groundbreaking research in that lab way back, all right? And in turn, there's all sorts of crazy things about the lab. Before we went up there, there were some other students that had been there a few years before, and they're like, oh, when you get there, make sure that you check out the moat around the facility. So I'm thinking we're going to see this giant castle, and there's going to be this moat around it. I'm like, what the hell is going on here, right? What kind of lab am I going to that's got a moat, right? Crazy. And are there like alligators or piranhas in it? What's going on, right? When we got to the lab, didn't even know that I crossed the moat, all right? It was like the curb, a little gutter in the curb, but it went down. It was a little moat, but it was about that big, all right? And about that deep, right? What the heck do you think you're going to keep out with a moat like that? Or keep in, right? Ticks, that's probably what, right? Maybe some other stuff, but ticks for sure. And that moat wasn't full of water. I'm sure it was full of mineral oil or oil, some sort of oil, because they don't like that. And either way, ticks from the outside or ticks on the inside aren't going to go one way or the other, keeping them intact. The moat was dry, it's empty. There's different techniques that they have now to ensure what they're dealing with stays where it should be, all right? Kind of crazy, all right? So we see all of these crazy things, and there's all sorts of different labs with all sorts of cool research happening. And we get into this one scientist's lab that looks pretty empty. There's a little bit of material around the edge. We learn it's a lot of the lasers that are focusing in on this machine that's the confocal microscope that looks like a lot of computers. There's a bunch of monitors and this big box. And this guy walks in, kind of mad scientist like, I felt like when he rolled in. I think it was to impress us. He's got his lab coat on and he's got two big ass syringes in his hands, right? I'm like, what is going on, right? This is interesting, right? And he asks us, there's just a few of us in this room. He asks us, we're all a bunch of undergraduates, right? How many of you guys have seen macrophages eating bacteria, right? None of us have seen that, right? But I know macrophages want to eat bacteria, right? In one syringe is some macrophage, and in the other syringe is some Staph aureus. Little gram-positive cocci is how Staph aureus looks. And a macrophage looks like a big kind of giant amoeba, sort of like these big blobs minus the pinkish color inside of them, right? So it looks like on the computer monitor, as he has two ports on there, he injects his syringes into these things. And up on the monitor, all of a sudden, here's a macrophage. And then there's a couple of little cells in clusters. That's the stack, all right? Bunch of little spheres on one end, and it looks like they are super far apart, but I bet they're, you know, a few centimeters from one another, probably, all right? Macrophages can smell organisms, and they move toward them by a chemotactic response. They sense them, right? And all of a sudden, the macrophage that's hanging out here minding its own business, you could tell it smelled there's a bad guy in here, right? And all of a sudden it starts pushing out its cytoplasm. It moves by pseudopods like an amoeba would move, right? Starts pushing out, projecting its body, if you will, and it pulls itself towards the Staph aureus. And it gets right up to the staph, and you see it surround these cells, and it pulls it to the inside, and it's like a little pump. Pulls it in, and it just phagocytized that. It just ate it. I was like, what? That is awesome, right? And I know there's definitely some videos that show some of that stuff. My mind was just boggled. I was like, you see images, you see, oh, here's a cartoon of something getting phagocytized, but to see it happen right before your eyes, you're just like, what? That's cool. That's my memory of confocal microscopy. It gives you opportunities to visualize stuff, real time, living things. You don't have to fix it to a slide to look at it. You can see it happening right as you go. This microscope is an electron microscope. There's two types of electron microscopes, transmission and scanning. The transmission is more of a kind of see-through sort of an idea, giving you details about maybe some insides of stuff, although you can also potentially see outside as well. And a scanning is more to give you surface stuff. In both instances, you're gonna, you need to stain up these things. Transmission microscopy depends on really what you're looking at. It might be thin slices of something that you're looking at some details with. There are other potentials, kind of just depends on what it is, all right? The scanning can be large things. There's all sorts of different images you could search up with microscopy things and some scanning electron microscopy images. There are some brilliant images that you're just like, oh my gosh, awesome. And some other things that might be flat out terrifying when you see it. Who knew what the dust really looked like in your house? Do you want to know? Do you want to know what's truly in there? And all of a sudden, here's this gigantic looking dust mite that's feeding on your dead skin cells that's on the shelf. Awesome and a little terrifying, right? So we had in the biology building down in the basement, kind of by where your micro lab is, a transmission electron microscope, pretty old one, but it functions very well, all right? And it's one that probably a decade ago, there was a donor that helped to bring it a little more into the 21st century. With this, you've got little oculars, you could do stuff, you would take pictures. Now a lot of it is linked right up to computers, and those images get imported directly. Different, all right? And she in turn donated thousands of dollars to get this computerized. The microscope itself was like almost $500,000, right? Crazy expensive, right? And also, you know, the service contracts on some of these things are outrageous as well. To have somebody come in and look at it periodically, thousands of dollars. To work with the electron microscopes, $50,000 a year to have somebody come in and deal with it. I'm like, what the hell? Wouldn't that be a good job? Or maybe not, right? Probably not, right? So we have one of these. This is something that I knew we had. I saw it, but I never used it, right? I teach a course called C phages where we look at basically like virus hunters. Look for bacteriophage, which is a virus that wants to infect a bacterium. They don't cause issues with us, all right? And can you find it in the environment? Can we cultivate it? Can we get it to grow up to large numbers? And then can we visualize it? We stain these viruses. We actually come down to the chemistry or the physics department. And down in the basement of that building, in the physical science building, there's a bunch of different labs, and one of them is a radiation lab, because we stain our virus with uranium. And as soon as I'm like, I don't think I want to do that, right? Uranium doesn't seem like something I want to play with, right? It makes me think of The Simpsons, right? I think that's plutonium, but or whatever it is. It's probably uranium, right? Whatever he's got, in the back, right? I'm like, that's not, I don't want that. It's very low issue, but we still don't deal with it. Somebody else does, that's all suited up and has the Geiger counter testing themselves. So the viruses, all of the sudden, that are invisible, you can't see even with our regular microscopes, show up. And they're a few, maybe 100 nanometers long, maybe a little bit less, all right? Very, very little, all right? And the transmission is the one that we have used. I'll see if I can, I know I've got an image of some of those. I'll see if I can get one and post it on Moodle so you can see how some of them look. We'll show you some pictures of them in here, but so last year was my first time. I've known it's been there for forever, but I've never got to actually deal with it. And we got to see them on there. The technicians were initially finding stuff. And once they found it, they stepped back and they let everybody kind of get a feel for it and turn the dials and move the stuff around and figure some stuff out. And I was just like, what? I get a turn? Like a kid in the candy store. I was just like, this is awesome. All right. Pretty cool. All right. So high energy electron beams. It takes a bit of time to get this machine up and going and get all of the stuff working properly. There's a big tungsten filament that gets super energized, and electron beams get focused down on the specimen that's mounted on this little like vise that goes right into the machine, and you focus in on it. And it's a little teeny screen that the specimen is coated on, right? That's what we're seeing in here. And it shows up on that screen, right? So similar idea with that. So in this case, my first thought as I see this, I'm like, oh, that looks like mitochondria. That's what I want it to be, but it's not. This is more biofilm stuff, not the same one we saw before, but similar sort of an idea with multiple players. This next image, and oftentimes when we show electron microscopy images, there's some coloring in there. It's like artificial coloring. They really aren't purple when we're doing this stuff with the microscope. It's more black and white that you're really getting, all right? So they have color enhanced this stuff. The Staphylococcus is a gram-positive bacteria, so it should look purple, and they've colored them purple to kind of enhance that a little bit. But really, they'd just be dark spheres is what we would be seeing. In the background, this is the screen that the whatever it is, in this case, the bacteria has been placed on, and then there are substances coated on it, probably uranium in this case, to make it be visualized. It would look like this, right? And then they go in with a computer and all of that dark stuff, they just turn purple or whatever else. So there's little bits of pores in the screen that you can see. That's what this is, right? And then the bacteria on the surface. Huge compared to how we'd look at them with a compound microscope, all right? And we also say they look like clusters of grapes. And here I think is a great image of that. They look like some grapes, right? So biofilms, we've mentioned the littlest bit, and we'll talk more details about some of that stuff. So here we can see some of the formation of that. And there's definitely some differences between the free living or free floating planktonic bacteria versus those that have come together to form this sort of community. There's a lot of sugary coating like that glycan that we saw in that other image that helps to kind of provide a glue to get everybody to kind of interact and stick together, right? Oftentimes there are multiple participants in a biofilm where they cohabitate and share resources, and that sets the stage for these interactions. When it comes to things like in your body, biofilms can be an issue. It's hard to deal with and treat those situations because they're, you know, kind of glued in there and encased in that as well. So to get it to move through there is a bit of a problem. And this, we're looking at Pseudomonas on some surface is what we're seeing there. So here's some fluorescence staining, and this is on some stainless steel biofilm growing on that, which I think is similar to what that's really trying to show us. And this DAPI is a type of fluorescent stain that's allowing us to visualize that. And we can see the sugary coatings and the bacteria participating in that. So there are numerous things, and these tables show those specific ways that we may be doing it. It's not all the same images, but what we might be able to see. So our bright field, like I said, the way that we'll see stuff in the labs, right? Dark field also could be the same, right? With just an addition to that. And then the others, the phase contrast. Here's the DIC, differential interference contrast. It gives you this kind of surface image of what you're seeing. Fluorescence confocal and the 2-photon, we didn't talk about that, but it's similar, kind of like these electron microscopy images. So transmission and scanning here, this is some Ebola. I don't want to see that in the lab, but a different virus. And these are more of a filamentous virus. And then Campylobacter jejuni with the scanning, giving you kind of a sort of three-dimensional sort of idea with them. Campylobacter is, there are a handful of bacteria in milk that are bad news and definitely can cause some serious issues. But Campylobacter is kind of top of the list for bad intestinal issues. And one of the main reasons why milk gets pasteurized is to deal with Campylobacter, right? You don't want it, all right? It's, you got that going on. Lastly, with these guys, the scanning and the atomic force allowing us to zoom in. Here we're looking at a couple of different substances. One was cellulose, carboxymethylated nanocellulose, and gold, as we see that. Okay, we almost finished all of this. Just the last little hint of stuff, but I'm going to quit right there. I know it's quitting time. So have a great weekend. We will pick up with this and start 3 on Monday. Good day to all. Have a good weekend. First exam is going, I believe, opens next Friday.